Thank you so much, everyone, for joining us today. Hello to everyone. My name is Cynthia Henson, and I'm with SFU's Faculty of Science. I look after the Outreach and Engagement Program on behalf of the Faculty of Science. Thank you so much for coming in today and joining us. This is SFU's Cafe Scientifique, and this is a discussion series hosted by the Faculty of Science. And what this is we run this series to engage the community in meaningful conversations, to get to meet our research faculty and to discuss relevant topics in science. We're happy to have you with us today. We welcome both those who are joining us throughout the series, who have joined us in our past sessions, as well as all those who are joining us for the first time. Thank you so much for coming in today via Zoom. We thought of doing this virtually for obvious reasons, for all these because of all these distancing restrictions, and I hope all of you are comfortable in running through a Zoom call such as this. Before I go ahead and introduce our presenter for today, I'd like to acknowledge two of our SFU colleagues, Dr. Lina and Amir as well. Both of them are, well, they kindly agreed to helping us with a Q&A bit of this session today. They both work in the same lab as our presenter, and they know much more than I do about the Mango project, which we will be hearing more about today. So thank you so much, Lina and Amir, for helping us with this session today. For those of you with questions, there are two ways of doing this. You can either type in your question on the chat page, or you can unmute yourself. Sorry, before you unmute yourself, you can use the raise your hand function. For those of you who are familiar with Zoom features, that's just in the lower side of your screen. If you see manage participants, if you click on that, there's a raise hand function. You can just click on that and then unmute yourself and say your question. So either typing it in the chat page or raising your hand will work. I should say that we are recording this session also, but we're just recording our presenter's presentation, his slides and his speaking part. That recording will be uploaded on our website after tonight's session. Also, I should mention that after this event, we will be passing on an online feedback link via email to all those who have joined us today. We would appreciate your comments. We're doing this virtually for the first time, so your feedback, any comments or suggestions that you'd like to share will be well received. We'd also like to find out how you found tonight's discussion. Anyway, on to the main part of this program. It's not me, it's all about the presenter. SFU's research community is engaging and innovating to respond to the COVID-19 outbreak. In today's session, SFU Faculty of Science Professor Dr. Peter Unrau from the Department of Molecular Biology and Biochemistry talks about Mango, a state-of-the-art imaging technology that's also being used within coronavirus testing kits and the potential uses in detecting other viruses such as seasonal flu. I'm thrilled to have Peter with us today. Thank you so much, Peter. Friends, please help me welcome Dr. Peter Unrau. Peter, over to you.
Well, thank you very much, Cynthia. Can everybody hear me? I think everybody hopefully can hear me. Let Cynthia know if you can't, but I'm going to assume that you can. So we're in a different world at the moment, and I guess this is one that I never really anticipated encountering. I mean, I go to the grocery store and I'm 2 meters apart from somebody. I can't buy any of the the natural food products because they're all covered with plastic. Airline flights are shut down. This is really quite unprecedented. And so I am not a viral researcher. I'm an RNA biochemist, and we've been developing in my laboratory some technology to look inside cells and image RNA within cells. But as a result of this COVID-19 outbreak, we've been refocusing in the last months on how to address viral testing for the COVID-19 outbreak. And so I'm going to tell you a short story. I'd be delighted if you interrupted me during this presentation if there's anything unclear, or if you just have a question. I think that's a more logical way to go through a presentation like this. So please do ask as I'm speaking. But I'm going to talk about what really is the issue here and what my laboratory has developed before this viral outbreak that we're trying to fit into the COVID-19 testing regime. All right, so this is my lab on the screen. It's a fairly small unit, and this is before the outbreak. We're all close together, and I will talk about some of the people that have been doing the work here, but particularly Lena and Amir have been working on the COVID-19 research that I'm going to talk to you about. Okay, so let me get to a next slide here. This is the situation as a couple of days ago, and there's a number of sites that report the number of cases and the number of deaths. Right, so we're about to pass about 5 million cases of people that have been diagnosed with COVID-19 around the world. And of those 5 million people that have been diagnosed, about 500,000 of them have actually died. So about 10% of the people that have been diagnosed with COVID-19 have actually died. So there's a couple of very misleading things about these plots, because the only reason that we know how to plot the total number of cases is by actually diagnosing correctly a person with COVID-19. So almost certainly the number of people that actually have COVID-19 and didn't actually have severe symptoms and didn't actually report to a doctor is much higher than the number that we're showing here on this plot. But still, these are alarmingly high numbers. This is about the number of people that die each year from the flu over a year, and this number of people have died over, well, since January of this year have already died of COVID-19, right? And the total number of cases here, it's not that much of the population, which is equally alarming, right? So this is about 0.1% of the world population has been diagnosed with COVID-19. So there's a lot of people that could still get the COVID-19 virus. All right, so this is the big picture. There's a couple of things that are interesting about these curves. This is plotted as a logarithmic curve, but the initial outbreak looked like it might have plateaued by sort of the end of February. This was just viral outbreak from the initial cases in China. And now we're seeing a second plateau right now. So the real concern, I think, that most of society has is trying to make sure that this, in fact, actually stays flat and doesn't actually inflect up again now due to a relaxation of all of the restrictions that have been imposed on society as a result of this virus. There's a lot of people left to infect. We don't have any vaccine against this particular virus. This type of virus, such as a coronavirus, is particularly hard to get vaccines for, so this may take a while. And so this could easily trend upwards if we're not careful with our social distancing that we're trying to practice. And unfortunately, if this trends upwards, this death curve is going to trend upwards almost in direct proportion. It's about 10% of the population that get this virus seem to be dying, right? But you get diagnosed if you're actually quite sick, which means that you've gone to a hospital or you've consulted some medical authority and they have decided that you should have a test, and you take the test and it comes back positive. So a critical concern right now is how many tests can we actually give to people, and we need really cheap and reliable tests. We've had cheap tests that are not reliable, we've had reliable tests that are not cheap, but how do we get a cheap and reliable test? And so I'll try to address some of those points as I go through this talk. All right, so this is in Canada. The numbers are obviously much smaller, the population of Canada being only about 37 million, but we're typically trending around 1,500 people being diagnosed with COVID right now. And of those, about 10% of those are dying. All right, and you might imagine that we're trending downwards right now, but we've seen sort of oscillations in the data. The statistics are not relatively large, right? So we need to be trending these numbers down. And maybe they are coming down right now. BC is actually doing pretty well. We don't have too many cases coming in at the moment to our hospitals, but it can easily go up rather than down. And so these numbers are all in a state of flux. All right, so what is COVID-19? It's a coronavirus. Coronaviruses are actually really quite abundant. One form of the common cold is a coronavirus. COVID-19 is a coronavirus. Coronaviruses infect poultry, pigs, mammals like bats. They're quite a complex. They're large genome viruses, and they're hard to treat. We don't actually have a vaccine for the common cold, and that's not for the want of trying. The common cold doesn't typically kill you, but it has been hard to get vaccines for these types of viruses. So this is what it would look like in a model representation. It's a membrane virus which has surface proteins. This large triangular red blob, obviously it's not going to be red in the real virus, but interacts with proteins on the surface of your cells, and that causes this virus to actually be brought inside of the cytoplasm of a eukaryotic cell, your cells, actually allowing the genome of this virus to go into the cytoplasm of your cell. This is the nucleus of a cell, which is where your DNA, your chromosomes are. Normally the information is flowing out of the nucleus, but this virus is putting information into your cells, information that's required to copy this virus and make more of these guys so they can go off and infect. To give you a sense of scale, this guy, if this was like a cantaloupe, which is a typical human cell, so imagine a typical human cell being a cantaloupe, this guy would be about the size of a grain of sand. All right, so they're small, but they're not, they're relatively small compared to the size of the cell. Okay, this is actually virions actually coming out of, blebbing out of the cytoplasm of a cell. This is an electron micrograph. So here are actual individual viral particles coming out. And these are blebbing out into the space outside of the cells. If they're in your respiratory tract, which this guy's in your respiratory tract, if you sneeze or you cough on somebody, these viral particles are going to fly out and infect another person, which is why wearing a mask is such a good idea. The mask not really protecting you, but protecting others from you, right? It has a minimal effect on protecting yourself, but it really does cut down on the particles leaving your body, largely from you coughing or sneezing. All right, so back to this cantaloupe or this eukaryotic human cell. Normally DNA is in the nucleus. This is your genome. It controls the cell by transcribing the DNA genome into RNA. RNA then leaves the nucleus through little pores in the nuclear envelope. And that RNA goes into the cytoplasm where it actually starts being converted from a messenger RNA into an actual protein that actually does some job inside the cell. So this is the command center in the center, the nucleus. And you're making all of the machinery in the cytoplasm. COVID-19 is just really tricking the cell by injecting an RNA into the cytoplasm that looks exactly like actually an RNA that the nucleus would make. So it really tricks the cytoplasmic machinery into believing that that RNA that the virus has is a messenger RNA that came from the nucleus and therefore should be processed. Only problem is that it's being processed into viral machinery, not cellular machinery, and therefore the virus succeeds and the cell loses out in the end. Okay, so you can compare these coronaviruses. I've mentioned that common coronaviruses are things like the common cold. These are shown in green here. What we're looking at here is like a family tree. We're comparing the distance between the viral genomic sequences. So if you're on a horizontal bar like that, you're quite closely related. So these two guys are closely related. These two guys are closely related. These are more distant. And these are the guys that kill you. So MERS is something that you might have heard about. SARS was a really bad virus. It killed you if you got it, but that it didn't spread. And unfortunately, the COVID-19, which is here, spreads. It doesn't kill you as much as SARS, but it spreads, and therefore more people get infected. So therefore, in aggregate, more humans are dying from this COVID-19 than from SARS, which is actually more lethal if you get it. The interesting thing here is that when Amir made this tree, he also put in a bat coronavirus. It's actually quite closely linked in terms of its sequence to COVID-19. And so that probably traces some route back to where this virus actually came from. So there's coronaviruses all over in primates and in mammals. And so a bat's a mammal. And so pigs are on here someplace too, and poultry slightly further away. So we've lived with coronaviruses for a long time. They're miserable. We're currently dealing with a miserable one that actually tends to kill us. So we got to deal with this one way or the other. All right, so how do you know currently if you have COVID-19? The testing is actually fairly barbaric. And it's because of SARS actually that we have this testing. This is what would happen if you were going to have a test. Maybe you've already had a test and can relate to me directly about the test. But this is what you do this is what would happen if you report to the emergency room. They're going to stick a swab up your nose, and not just a little ways up your nose, all the way to the back of your sinus, right? So this is quite painful apparently, and awkward. So you take the swab, you're going to scrape off the cells and mucus that contain the viral particles, and the swab comes out. Okay, so now this swab, once it's coming back out, is going to have the viral particles on it. So what actually happens next, it's going to be put into what's called transport media. This actually keeps the virus alive, which is quite remarkable to me, actually. It doesn't kill it, and that's actually because in times when you don't have a pandemic, you have to have a test where you can actually culture this thing, and you want to keep the virus alive. But all of this is because we've had a previous experience with a small outbreak, nowhere near the scale of what we're dealing with now. So all of this pipeline that I'm explaining to you is not optimal, really. It's just what we have and what we have to use, because we can't change these guidelines really quickly. Otherwise, we might not be sure anymore that we actually have a positive test. So it's because of the history of these tests that we have what I'm just about to describe. So that swab, which is inside this tube, is then shipped to a testing facility. This is quite a dedicated facility. It's got quite a few expensive pieces of machinery in it, right? So the machine at the end of this that's actually going to do the RT-PCR test is probably 50K, 100K, one of these units down here. It's probably got several of those in it. It's got numerous technicians that are pipetting a lot of liquids. So they have to take the sample, they have to extract the RNA from it. They're then going to do an RT-PCR assay in a machine. They're going to get the answer from start to finish in probably about an hour and a half, something like that. But suppose that you live in a rural community and you get tested, it has to be shipped to the center. That's a couple of hours or half a day or whatever. Once it's received, a couple of hours, you're going to get your answer back inside of 24 hours if you're an essential service person. But if you're non-essential, you're going to get the hour longer than 24 hours. So it's quite an elaborate pathway. It works. It's very reliable. It's very structured, and it's very outdated. But the key ideas is that we're working in these tests with actually taking the RNA out of the sample of the virus, converting it into DNA, and that DNA, that's done by an enzyme, is then amplified by what's called the polymerase chain reaction, which exponentially amplifies a segment of the DNA that you've recovered from this reverse transcription step. And because you've made a lot of DNA, you can see a signal, a fluorescent signal inside this machine that reports either a positive, if there was an exponential rise in this signal, that means that there was viral RNA there, or you don't see anything, in which case it's a negative. So this pipeline exists. It requires a lot of people. They have to be technically competent. And the expensive machinery just means that you can't have very many of these test facilities. So it'd be great if you could relax this collection of the sample to make it as simple as possible. And that's what a lot of people are thinking about. And that's what we started thinking about in February. So this is my lab now. We're 2 meters apart. We're all holding our ethanol squirt bottles so we can sterilize things. And while there is a pandemic, we're actually a pretty cohesive team and we're having a lot of fun. So everybody's smiling. But it is different. It is very different. The university is shut down and we're all by ourselves, roughly speaking, up on SFU campus. So it's been very different. All right, so let me tell you a little bit about what we did before the pandemic and how we're using that technology to try to address making a new test that hopefully will be more efficient than what's currently being done. So for a long time, and by long I mean since before the turn of the century, in the 90s, people have been able to study proteins in cells, and they did that by using fluorescent proteins that exist in nature. So a lot of jellyfish have fluorescent proteins in them. So here's a jellyfish that's glowing in a fluorescent way. So fluorescence just means that if you shine a high energy light source, a high energy photons onto a fluorescent protein, it emits lower energy photons. So here's the light that you would see with your eye if you just looked at it with white light. But if you put it in a dark room and shine short wavelength light on it, this is a light that's actually coming out of the tube, making it glow, sort of like a glow stick, where the chemical, except that instead of the chemical energy causing the light to come out of a glow stick, it's actually wavelengths of light that are more energetic. All right, so what people have done is made a whole bunch of fluorescent proteins of different colors, starting from the naturally occurring fluorescent proteins, and they've put them into proteins that exist within cells to sort of see what's going on inside of the cell. A key problem is that RNAs are not fluorescent, so you can't track RNAs the same way as you track proteins. And so we decided that we should try to solve that problem by making aptamers bind to fluorogenic dyes. These fluorogenic dyes could get inside the cell. They're not particularly fluorescent by themselves, but when they bind to the aptamers, they could become very, very bright, and in this way look like a fluorescent protein. So if you have a fluorescent protein, you could tag something like actin. This is 2 cells. There's 2 nuclei here that you can't see because there's no actin inside the nuclei. But these filaments are providing structural elements of the cell, and it's just a particularly nice image that I took here to show you. But you can see the protein filaments that are providing the structure through their fluorescence when you put them inside a fluorescent microscope. The microscope excites the GFP, and the GFP emits, in this case, green light, and you can see where the molecules are inside the cell, making it a very useful technique. And so it has many applications. So if you have a protein that's involved in disease and it mislocalizes to another part of the cell, you can see that in a microscope, and you can understand why that disease is actually happening by the microscopy that actually correlates with its mislocalization. All right, so what Lena in the lab selected was a set of aptamers that we call RNA Mango, and these RNA Mango aptamers are fluorescent when they bind a fluorogenic dye. And so these are just tubes of different fluorophores bound to the aptamer glowing. So you can't actually see the excitation light. It's not visible to the camera that took this picture, but this is just light coming out of the tubes from the excitation light actually exciting the fluorophores that are in bulk in these tubes. And these are just the names of the compounds that are bound to each aptamer, which are not particularly important. So we're just hoping to, before this pandemic, to try to have a routine way of imaging RNAs inside cells, and therefore, in analogy to how you study proteins inside cells, which have many disease ramifications, we'd be able to study RNA in the same way. And since RNA has many disease ramifications, we'd be able to make more progress in studying human health problems. So this is just a sort of big picture idea. So if you could actually do this in live cells, which you can do with proteins and which we've been trying to do with RNAs. It's sort of analogous to tracking whales, right? So here's a guy putting a beacon onto a whale. So you can think of this as RNA Mango, and this is the RNA. It's a whale, and it's a GPS beacon with a transponder on it, right? And so a satellite will detect this, and you can see where the whale is as it migrates from, I guess, its breeding grounds to its feeding ground. You can see where each whale goes. You can say, okay, well, these whales do all this stuff. And if you're a whale biologist, this is fascinating, and you know where the whale is, and you can see that they go in particular routes. Some of them stop in odd spots. Sometimes the beacon falls off, whatever. But you learn about whale migration in this way. So tagging RNAs, just like tagging proteins, allows you to study the migration of an RNA inside a cell. And if it's anomalous in some way, maybe due to a disease, you can learn about it, just like you could learn, a whale start migrating differently. All right, so just to give you some idea, this is maybe a bit technical, but these aptamers get very bright when you add a fluorophore to them. Or conversely, if you have a fluorophore in a tube, so this is a tube with fluorophore in it, you can't see very much. But when you add the RNA, the aptamer that binds to the fluorophore, it gets very fluorescent. And it gets thousands of times more fluorescent as a result. All right, so these are just two different fluorophores in the tube by themselves. They're not glowing very much, but when they bind to the aptamer, it becomes very bright. So you have to have both together for it to be bright. Obviously, the RNA is going to be the tag that you put into the RNA that you want to track, and the fluorophore can just soak into the cells. All right, and they bind very tightly. They have nanomolar binding affinities, which means technically that they're basically covalently bonded together inside the cell. And so quite literally, this is what an instrument that's sensitive to fluorescence actually sees when you put the fluorophore in to the instrument. When you actually put the RNA in there, you suddenly see a dramatic signal, which is directly analogous to what you would see with your eye. This is just the spectral properties that you would see in the instrument and gives you the exact wavelengths of the excitation. In this case, it's about 530 nanometers is the emitted light from this particular fluorophore. All right, so they bind the fluorophore tightly. When they do bind it, it's very bright. And that allows you to see things. So one thing you can do is you can look inside cells now. Right, there's a number of technologies to do this, but the Mango technology works really well. What we're looking at here in green is tier 1 staining of actin RNA. Actin RNA is the RNA that would actually make that actin filament that I showed you in GFP before. But what's interesting is that the RNA that's made in the nucleus actually goes to the edge of the cytoplasm, and you can see the localization of the actin RNA there, where it's actually turned into actin protein, and which was the green signal in the GFP that I showed you earlier. And you can do super resolution imaging with it, which is technically quite interesting and opens up a lot of doors. So there's a lot of stuff that we can do with this, all of it inside of a cell. But the most interesting thing that we can do is actually see inside living cells, individual molecules of RNA. So what you can see here is actually one of the aptamers. One of our collaborators actually solved the crystal structure of it. It's a very small sequence of RNA, and this is its three-dimensional shape. You can see the dye actually bound up here, and that's its bound form where it's super bright. And it's got a very biologically compatible shape. It looks like an RNA helix, which is easily substituted inside RNA structures. So this is what it looks like at the molecular level. This is what it looks like inside a microscope. So we're just tracking an individual molecule of RNA that's being tagged with this sequence. So inside the linear sequence of the RNA, we put one of these tags, actually a set of them, and we're comparing it to a really competitive, well, the best technology out there. And we're seeing RNA very clearly at the single molecule level. So there's a lot we can do with this. And we were quite happy to charge ahead with this in January. But with the COVID-19 outbreak, we decided that we should focus on how to use these really bright aptamers to actually just detect COVID-19 from actual patient samples. So before I actually explain the COVID-19 detection, I'm just going to tell you how Lena actually did the selection. She, as a graduate student, did all of this work finding the aptamers, which was a huge battle. And then 3 guys from 3 different labs, Sunny from my lab, Alexei from Michael's lab at Strasbourg, and Adam from David's lab at Imperial College, and Bob from NIH all worked together. He did the crystallography. Sunny worked with Lena and characterized the aptamers in my lab. Alexei did some of the selection after Lena, and Adam did all of the imaging that I just showed you. So it's been quite a team effort after Lena gave rise to all of this stuff. Okay, so I got to describe a concept here of how you get a 3-dimensional shape out of an RNA polymer. So you want to find functional RNAs that can stick to these fluorophores. And so how can you possibly do that? It's not like we're engineers. We can't build an RNA that can stick to this fluorophore by design. We're just not smart enough to do that. So the solution that has been hit upon in biochemistry is the concept of in vitro selection. Start with a very large library of sequences, and then find the ones that are functional out of the very large and random sequence population. So basically you build all of the shapes, and then you ask which of the shapes stick to the fluorophore. An engineer would think that's a really stupid idea, because they would just say, well, let's build the shape that can actually stick to the fluorophore. And obviously we'd like to do that. We just can't figure out how to build molecules that way yet. People are making progress that way, but not with nearly enough sophistication at this point in time to solve this kind of problem. So we start with a very large library of shapes of RNA, sort of like antibodies that your body would build, and then say, well, which ones are actually able to stick to the target that we're interested in. And to give you some sense of scale, I said before that a cantaloupe was a human cell and the virus is about a grain of sand. Well, picture this Rogers arena filled completely with sand. Each one of these pieces of sand is a different shape of RNA. It's a small piece of RNA that each one has a different shape. And then just pretend that, let's say, 1 or 2 of them are actually functional. Your job is to actually go through this arena and actually find the functional grain of sand, right? So you can pretend that, all of the grains of sand in here are just white beach sand, and you have 2 red grains of sand in there someplace, and you've got to find them. So that's the goal of the project. But it's biochemistry. So you've got a tube, and you put 10 to the 15 grains of sand, which are actually RNA molecules now, which are much smaller than a virus, into that tube, and you're asking which are the 2 molecules in this tube that actually are functional, that combine to that fluorophore. And so the beauty of nucleic acid is that you can copy it. While that is beautiful and that's what keeps us alive, it's also how the virus is copying and infecting so many people. So it has its downsides as well. But you make this library of around 10 to the 15 pieces of RNA, and then you design a biochemical step where the functional ones you keep and the inactive ones you throw away. And you keep the active ones, and you throw away the inactive ones, and then you can amplify the nucleic acid. So I just described how the COVID-19 test works. You convert RNA into DNA and then amplify it. That's what Lena did here. She did exactly that step, and then took that DNA, put it back into RNA, selected again for things that could bind to the fluorophore, and did this iteratively. So each time she did one of these cycles, there were less and less sequences left, but more and more copies of each sequence. Very much like biology, but very much in vitro and very much in a tube and in a lab. So all of these things she had complete control over. So she ended up with a tube of sequences that all could bind to the fluorophore, which was good. She had a large number of them, hundreds of sequences that could bind, in fact. The next step was to figure out which ones were really bright. So this is what Alexei did in Strasbourg. He took the pool that Lena had made, and inside each of so what you're looking at here is a microfluidics device. Inside this channel, there's fluid moving down, and it can go up and it can go down. Inside each one of these droplets, there's RNA, and it's one of the types of the RNA that Lena selected. And if it actually is very bright, it will go up. And if it's dim, it will go down. So it's a sorter. And it sorts by applying an electric field here that distorts the shape of the droplet. So it can go up if you turn on the electric field, otherwise it will go down. So you can see that the ones that aren't glowing, so it's glowing here and they're going up, and the ones that aren't are going down. Oh, I think I'm interfering with its playing. Yeah, so you can see it flash. When it flashes, they go up. When it doesn't flash, they go down. Right, so what's happening here is there's a laser shining through the droplets. There's an electronic circuit that decides, is it glowing? If it is glowing, then it turns on the electric field and it sorts it to go up. So in this way, they could sort through all of the high binding affinity aptamers that Lena had found. So Lena took the problem from 10 to the 15 down to about 10, 10 to 100. And Alexei sorted through maybe 100,000 droplets, a much smaller number of RNAs to get the best, brightest ones, which is where the project ended up at around the time of the COVID outbreak. So at the same time, Amir in the lab had been using this technology to actually do amplification to detect pathogens, actually, which was a curious coincidence in our case. But we've been working with a small company called Applied Biological Materials. Their issue was they wanted to sell a kit that could detect pathogens in tissue culture. So tissue culture is an experimental system that scientists use to, well, for many things, but you take typically human cells, you grow them in a dish, and you do something to them. Your experiment might be to see, well, is this compound good against cancer or of a particular type? So you have a particular type of tissue culture cells and you're working with these cells, but they can become contaminated. And if they become contaminated, your experiments are useless to you because the cells won't act normally anymore. So it's important to know if they're contaminated. And so Amir was working on this to see if he could rapidly detect bacterial contamination in tissue culture. And if we could do that, we could sell a kit with ABM and that would be fun. So he developed this isothermal methodology using mangoes. And I can explain it briefly. Maybe you guys will have to ask questions for it. But basically, if there's an RNA sequence, which is coincidental, but is perfect for COVID-19 because it's got an RNA genome. If there is an RNA sequence you want to detect, isothermal amplification is like PCR. It takes two primers. These are two pieces of DNA nucleic acid and a set of enzymes, and it amplifies a segment of this RNA between these two spots. So this line just means a piece of RNA. This line here means a piece of DNA that has the same sequence as this red guy. And this guy has the same sequence, but a little bit of a trick. It's backwards, and the sequence is what's called the reverse complement. So if there's A, C, Gs, and Us in RNA here, this has the reverse one that's complementary to it. So an A pairs with a U and a C pairs with a G. So it allows amplification from here to here. And you can do that with 3 enzymes, and you can do it at a single temperature, which is very convenient because your machine to do this becomes a lot cheaper. And what Amir found was that if he did this twice, if he did it with one set of primers and then took that reaction and put it into an inner reaction with 2 primers that are inside the outer ones, and if in addition he put one of the Mango aptamer sequences here, he could detect very, very low levels of templates. So what he's showing here in this graph is that if you put nothing in, there's no amplification. And if you put in 150 molecules per microliter, you get a robust signal for either of the templates, either the blue one, which is E. coli amplification, or the green one, which is another bacteria. Both of them amplify. But if you use one of the primer sets from, let's say, E. coli with the other bacteria, you don't see a signal. So it's very specific. So you amplify what you want to amplify, but not if there's the wrong target there, which is perfect because you don't want to amplify, a target that is not what the one that you're looking for. You want to actually shoot your arrow at a particular type of pathogen and say, well, ask the question, is that pathogen in the sample? And if the answer is yes, well, you know the answer. If it's not, it should be no. And then you can say, well, it's not that pathogen, it must be this pathogen. And so in this way, you could screen and identify which pathogen it is. But the remarkable thing is you could get down to about 1.5 molecules per microliter, which is not very many molecules, right? You can count that number. And so it's also very sensitive. So at this time, So he published this paper just before, just at the end of 2019. And then we were charging along on our research projects, and then this COVID-19 outbreak happened. So CIHR, which is the Canadian Institute for Health Research, recognized the pandemic as being of federal level importance, and they announced a competition, which they called the COVID-19 CIHR Rapid Response Fund. And they basically just asked researchers to try to solve this problem for them. So we wrote a grant, and the remarkable thing about this, grants normally are submitted, you normally take several months to write a grant. And then normally it takes several months to get the answer from writing the grant. And then you normally get the money, and then you normally work for a while on the grant, and then you publish your papers, and you move on with your life. This one was announced. A week later, they closed the competition, and a week after that, they announced who was funded. So, you know, blindingly fast for our federal program. So we put together a team of organic chemists. Rob Britton was part of that team to make those small fluorophores that I was showing you. We brought in a virologist, Masa Nakura, who's just about to get a CL3 biosafety facility up and running. So this level facility is where you could actually culture this COVID-19 virus safely on the SFU campus. And that's quite important for us because we need a supply of that virus to do our research properly. And there's a number of other researchers that will use this facility to test things like antibodies and small molecule inhibitors. In fact, Rob Britton has got some small molecule inhibitors that may well prevent the growth of the virus. There's also 2 clinicians that are at St. Paul's Hospital, joint with UBC, that are actually sticking swabs up people's noses. The good news is that there's far fewer positive people than we're expecting. But at St. Paul's so far, we've had 13 positives, 11 positives that we've collected so far since the start of this study. We were expecting probably 10 times more than that based on the initial data, but that sort of plateau that you saw in the data that I showed you earlier really has had its effect. And in BC, it's been particularly effective, but we do have actual real samples, which is quite useful because those are authentic samples. But from our point of view, we wanted more samples. From society's point of view, this is really good. But we've been working with Don and Mark as to getting more of these samples. And then finally, we have an engineering team that's trying to build a prototype device to test the kit that we're trying to develop. And so it's been a lot of fun talking between all of these teams. We had our first group meeting between all of these members just last week. And there's a lot of progress that's happening, a lot of progress that is remaining to be done. And that's what I'm going to tell you about in the next couple of slides. Okay, so this is the assay actually that Amir developed working on a COVID-19 RNA. And so what you see is a fluorescent signal detecting easily an attomolar of concentration, which is inside the tube is 12 molecules inside the tube. So at the top here, we're detecting a femtomolar, which is kind of boring. That corresponds to somebody, I should explain. So if you have this swab that you've collected and there's going to be a certain number of viral particles on them, countably many, right? I mean, there's a definite number of viral particles on this swab, but it's not that many. And so you have to actually be able to count or have an assay that's sensitive to very small number of viral RNA genomes. And so having sensitivity that is low, like this attomolar sensitivity, is actually quite important. And so a lot of the kits that have been having problems recently just are because the sensitivity is just not good enough. So we're quite happy with this. This is the inner reaction that I was talking to you about, sort of responding detectably, probably inside 10 minutes. So we think we have an assay that can respond in about 35 minutes because there's an outer reaction that has to take about 20 minutes to be done. But when you have 0 template in there, the assay doesn't produce a fluorescent signal. Well, it does, but after a long time. And as you go higher and higher, it eventually saturates, but that's okay because that tells you that you have a positive. What Amir has shown recently is that it really doesn't matter if you put other nucleic acids in there. So if you add other nucleic acids, the negative stays negative and the positives don't really alter. And what's really quite fascinating from my point of view is that we think that he's sensitive to individual molecules in the tube. So here's an experiment where we're working at a very low number of molecules. This is actually before the COVID-19 outbreak. So it's not actually done with the COVID RNA. But here he's actually detected one molecule, and the other 2 times he did it, there were no molecules in there. That's our hypothesis. Whereas if we use more molecules, here we did 3 experiments, 2 of them were positive and 1 was negative. And once you move higher in concentration, it's always positive. We've actually gotten better at doing our assays since 2019. So now when we have 12 molecules, which is this blue curve, it's a robust positive signal. So the assays just gotten better just because we've worked on it harder as a result of the COVID-19 assay. All right, so this is the core output as a fluorescent signal in a short period of time. And this is what it would look like. It's sped up a bit, but the total time that we're talking about here is about 12 minutes. So this would be a positive signal, that's a negative signal, and this is a signal emerging. All right, so that's actually what you would see pretty much with your eye. So this is actually just using a cell phone camera to image this, right? So I didn't want to play it for 12 minutes. You guys would get bored, but 12 minutes is not a long time. And this is certainly something you could see with your eye, and certainly what electronics could see inside a relatively inexpensive device. All right, so what are we thinking this could look like? So we're hoping, and one of the key problems that I was explaining before is the expensive instrumentation that's used at the moment. The RT-PCR that's being done is very reliable, but it requires technicians that are highly trained and expensive equipment. One way to get around that is to use a simpler assay, which our assay does get simpler. So the technical level of performance required by a person is still fairly high. You couldn't really do this at home, but a technical person, let's say at a hospital, could run this assay. Another innovation that we're trying hard to master, and I think we've made great strides in recently, is to have all of the reagents dried down. And that allows shipping of the test kit at room temperature. The RT-PCR kits that you saw in the box that I showed you in the earlier slide are shipped on dry ice. They would be shipped from the manufacturer on dry ice. So roughly speaking, at -80, you then put them in your -20 freezer in your laboratory, and then you would run in one of these test centers your RT-PCR. But that's with liquid reagents, right? But if you dry everything down, it's much more stable. You can get up to a couple of weeks of stability at room temperature. So if you ship them fairly rapidly, you can ship them to remote areas. And so we're working with a company in the United States, in addition to a company here, the Applied Biological Materials I mentioned before, to get a large quantity of these test kits prepared as lyophilized reagents. And so if we can get that, which I think we're close to, and if our engineers can make a suitable test device, this is a 3-dimensional representation of something that they're 3D printing at the moment. And they're getting data from, actually, which is interesting. The idea would be that you'd have your lyophilized reagents in these tubes. You would add the patient samples to them. Once they start to glow, there's some LEDs in here that are exciting them, and there's photodetectors in the same device that can detect the glow. It's hooked up to a computer here that actually both powers it and actually reads out the fluorescent signal. As a sort of fail-safe, you can look through the top and actually see if the tubes are glowing. So it's lower throughput than the kind of tests that are done in a center. But a single person in a remote location could easily run, each of these could be, there's 8 tubes in this strip. Every hour, you could probably run through 4 people, something like that. If you were in a remote town of 4,000 people, that's probably a reasonable number of people to test with one person in a hospital. They'd get the answer, the software on the computer would upload something to a federal center, and therefore we'd know what's going on in that remote center and whether there's a problem. That currently is an issue for the federal government. Remote testing is not really happening at the moment. All right, so with that, I'm going to stop. And if you guys have questions, mean, the sheer volume of kits that are required right now is huge. The United States has a huge request for test kits. Canada needs, well, I forget the exact numbers. Amir and Lena probably remember it, but a while back they were looking for 50,000 tests a week. I think it's actually gone up by a factor of 5 since that the number I just mentioned, which is 50,000. So there's a lot of testing that people want to do. We're just limited by the kinds of tests that are available right now and the speed that people can scale up. I mean, we're, as biologists that normally are doing research science, moving very quickly, but we need to move even faster. So there's a huge pressure to get this done as quickly as possible. So with that, I think I'll stop. These are the people that I mentioned throughout the project. Bob was the crystallographer. Adam was the person who at Imperial College was working with RNA Mango inside cells. Alexei took the project over from Lena when she selected for the high affinity binding Mango aptamers and did the sorting. This is the CIHR rapid task force that we've been working with that I mentioned. And these are the people working in my laboratory. These are the funding agencies that are making the work possible. So with that, I'd like to thank you very much for listening.